All right, so we've talked about osmosis and talked about how osmosis is the diffusion of water across a membrane. Well, generally when we use the word diffusion, we're talking about the movement of a solute across a membrane. So movement of solute across a membrane. So this is where instead of the water being the only thing that can move, this is where that stuff that's dissolved in the water can move. It's where salt can go across, or sugar is allowed through, or food coloring, or anything like that is allowed through the membrane. So this is different because the water will still, do its thing with osmosis, but this is where solute can move. So the substance that is dissolved is solute. Okay, so keep those two words straight in your mind. I heard quite a few of you discussing that or asking about that when y'all were last week when we were talking about osmosis. Solute is the stuff that's dissolved. Solvent is the stuff that does the dissolving. So the universal solvent is water because it's polar. Solute is whatever it can dissolve. So diffusion is also known as the process by which molecules spread from areas of high concentration to areas of low concentration to create an even distribution. So for instance, let's say that I have some perfume and I spray it right here. I'm going to put some little perfume particles. I spray it. Well, as soon as it comes out of the bottle, the concentration of that perfume is extremely strong right here in this spot. But it doesn't just stay right there. Eventually, somebody across the room, if there's enough sprayed, will be able to smell it. And the reason is that those molecules, they don't like to be, really crowded. So they're bouncing, and they bounce until they can fill up a space. Okay? So that is diffusion. This is diffusion through the air. Different particles diffuse through membranes. So you're going to have a paper on the next page that actually I'll just show you on this video, and then you can pause it and put it together. But that paper shows you all the different types of diffusion. If we did not have diffusion through our cells, then everything that we breathe in would never exchange. Everything that we eat, we would never get nutrients from our food. So all of this has to happen. So let's look at a couple of different types of diffusion. I'm going to show you on the paper as I go through these. Okay, so you'll have to pause and color in your paper and stuff like that, but I'm going to show you these as we move through. Okay, so first main type is called passive, and I'm going to, no I'm not, I was going to say I'm going to color code that, but so passive diffusion is if you're passive about something, you don't really care, you're not giving it any kind of thought or whatever, you're not putting any energy toward it. Okay, well, passive diffusion does not require energy because the solute moves down the concentration gradient. Now, just to remind you about how a concentration gradient works, think of a concentration gradient like a crowded elevator. Okay, this side over here would be a very full elevator. Okay, lots of solute is over on this side. If you are standing in an empty lobby and this is you, and you need to get into an elevator, if that elevator is full of people, it's gonna require you to use quite a bit of energy to get in there. Okay, so that would mean you were moving up a concentration gradient. You're going from where it's low concentration of people to a high concentration of people. Okay, so that's moving up the concentration gradient. Let's say, in a different example, that you are in a crowded elevator. okay? And you're going, coming down to the lobby. When that door opens, you're in this very crowded elevator. When the door opens, the lobby is pretty much empty. So you basically just fall out into the lobby. You do not have to use any energy to make you go from that crowded elevator into the lobby. So that's passive. You're moving from high concentration to low concentration. So you're moving down the concentration gradient. So that is passive diffusion, where you don't have to input any energy. Okay? So let's go back to where we were. So passive diffusion is where solute moves down the concentration gradient. It does not require ATP. We're going to talk about ATP, and that is cellular energy. Okay, so no cellular energy is required. So on the next page, what I want you to do is go ahead and set up, I know yours is, much more blank than mine, but I want you to have, let's see, 1, 2, 3 large different colors, and then this one is the small one is the same color, and have at least 3 of these large ones of the same color. What you're trying to set up is that for this solute, all this different stuff that's dissolved, this is hypertonic. And down here you have, the matching colors, but this is very hypotonic compared to this concentration outside the cell. So this is your extracellular space. This is inside the cell, your intracellular space. So what I want you to show you are a couple of different passive diffusions. And we're going to get to some of these facilitated diffusions here in just a minute, but I'll go ahead and show you right now. Simple diffusion is where a particle can just move through the cell membrane. It's allowed through. It's going from very high concentration, the hypertonic, to the hypotonic low concentration. So it's moving down the concentration gradient. It is a simple diffusion. Okay? Facilitated, and we can go ahead and just look at that on this page right now. Facilitated diffusion just means that something's helping it out. Okay, I am a facilitator of biology, which means that I just help somebody out to learn it. So facilitated diffusion is movement of solute that requires a protein or some other help. So something that requires help. Okay, now we're going to look right now at 2 of these that are passive, and I'm going to use the same color right here. These are passive facilitated. So if it's just a channel mediated, uses a protein channel. I kind of think of that like a door. If you can fit through it, you're welcome to go through it. So channel mediated is this one right here. So it's basically, if you can fit through, you're welcome to come through. It is passive as long as it's going from a high concentration or hypertonic to a hypotonic low concentration, moving down the concentration gradient. Carrier mediated is going to be our next example that I'll show you on the paper here in just a second. I think of this like a bank teller drive-through. It opens and closes because this is a protein, and we've talked about how their shape determines their function. It's shaped a certain way that allows this to come in. If it can bind, that makes the shape change, and it opens up and it releases it this way. This is carrier mediated. It does not require energy if it's moving down the concentration gradient. Okay, so carrier mediated uses a carrier protein, and I think about that like the bank at the drive-through. When it's open on her side on the inside of the bank, it's closed on the outside of the bank, and then she pushes the drawer through and it opens on your side, but it's closed on hers. So that's kind of how I think about the carrier mediated protein. Okay, so those are all examples of passive, moving down the concentration gradient, not requiring energy, and you can either just move through the cell membrane if it's allowed, or it can use a protein channel. Okay, so the other type of diffusion is called active. Let me find a different color. Use green. So active diffusion requires ATP, which is energy. okay, requires ATP. That is cellular energy, adenosine triphosphate. So this is where the solute moves up the concentration gradient, or it's really large and it's moving in or out of the cell. So let's look at a couple of those. So over here are our active or requiring energy types of diffusion. So don't forget that every time you're referring back to this concentration gradient. This creates this pressure that we talked about at the beginning of this section, where because this is hypertonic, you know, these things don't want to be crowded, so they're trying to get through if they're allowed. So this red over here is hypertonic compared to the bottom, and it's really large. So we're going to say, and if you put kind of up here, that this means ATP. It's just like a little lightning bolt or something. Not too good at drawing lightning bolts. But this is endocytosis, and endo means inside, cyto is cell, and osis is process. So it's the process of bringing, you know, a large molecule into the cell. It pinches off some cell membrane around it and brings it in. And then you'll have cell parts that we'll talk about soon that will break this down so that the cell can actually get to whatever that is. Okay? Opposite of that is exocytosis. Exo is out, cell, process. So it's the process of removing something from the cell and using a little bit of energy in the meantime. Okay, another one is phago, P-H-A-G-O, cytosis. Phago means to eat. Cyto is cell, osis is process. So it's like the process of cell eating. Your white blood cells do this. Amoebas do this to kind of just reach out and grab the food particles, usually is what they're doing for amoebas. White blood cells will reach out and grab around bacteria or viruses that they feel like are invading you and destroy them, bring them into the cell, and then you have something called a lysosome that breaks them down. Okay, so those are three different, like, large molecules moving through active transport, requiring some energy. And then the last one right here is active because, if you'll remember your concentration gradient over here on the far left side, this molecule is in high concentration on this outside part of the cell, in low concentration right here, but for some reason it's being made to go like into that crowded elevator. It's being made to go up the concentration gradient. So this molecule moving up the concentration gradient requires energy, and this is called a protein pump. It's forcing it into this crowded space. We will talk about active transport doing this when we talk about how we create ATP. Energy is made because of this action right here. Even though it's required to make that happen, it's kind of funny. But anyway, not a lot of energy is required. It makes a lot more, but this is one of those examples where it pumps it into this the higher concentration. So these three right here, endocytosis, exocytosis, and phagocytosis, are examples of active diffusion using a protein. See, now that's going to bug me that I'd messed up on my colors, so there you go. So active diffusion using active facilitated diffusion and passive facilitated diffusion. Okay, so diffusion is a little different because it is the movement of that solute. Okay, and then this picture, you'll just glue it on the back. This will be Osmosis and Diffusion, page 6.